Hey everyone, this is Liam Sanya from Scientist.com, and in this special episode of Industry Insights with Crown Bioscience, we're speaking with Dr. Natalia Bezina, head of Crown Bioscience's 3D ex vivo patient tissue platform, and Dr. Martin Hornsfeld, a senior scientist in oncology. So we've recently had a webinar with Natalia and Martin covering different research tools and methodologies used to assay the suppressive tumor microenvironment, or TME, for immuno-oncology research. A link to that webinar will be in the description here, but today we'll be talking about native and reconstituted TME assays, how they can help in the preparation for clinical trials, and what kinds of therapies in development would benefit the most from this kind of testing. Alrighty then, Martin and Natalia, thanks so much for joining us to chat today.
Thank you. Happy to be here.
Thanks, Liam. Thanks for having us.
Awesome. So we'll cut right to the chase here. So in the webinar, we asked a number of polling questions about what kinds of approaches are most suitable for their purposes, as in, 2D or 3D cultures, ex vivo tissue, that kind of thing. We also asked what kind of immune cells are most relevant to their work. And also what kind of tumor indications are most relevant for the research in the context of the TME assay. So to start off with, what can you tell us about the polling results?
I was actually a bit surprised that almost 50% of the people were still voting for 2D cell cultures or organoid monocultures, while we have been trying to convince people, I think, through the course of these lectures to go for more complex setups. I was actually, I mean, fair enough. There's many good reasons to go for 2D and 3D monoculture still, but I do think it was an interesting outcome if you look at the interest in immuno-oncology, which is almost always more than just one culture, I would say.
Yeah, I agree with Martin. I was also surprised to see 25% of people still using 2D assays. But I think, it takes time for the field to develop, and 3D assays is still relatively new field, and it's still developing, and especially complex 3D assays, not only organoids, but the assays that have multiple cell types included, like what Martin and I work with. It takes time for the field also to accept them and to find the most relevant assay. And it was also interesting to see what cell types people were thinking if they would use these complex assays were the most relevant ones. And I think the majority were still T cells, right, and lymphocytes. But there were some other interesting cells. What do you think, Martin?
For sure. In the end, we forgot, of course, the one that everybody missed, the option fibroblasts. That was, of course, why everybody was tuning in for the cancer immunity cycle talks. But no, for sure, it's. I think it's nice to see that there's a broad interest also in different myeloid populations. Of course, quite a complicated field, up and coming if it comes to immunotherapy approaches. T cells, of course, are already a decade long one of the favorites to study. So no surprise there that most people are interested in working with those. Not so much dendritic interest, so for dendritic cells, although I think there have been quite some promising studies recently where you can condition dendritic cells to start presenting tumor neoantigens, things, and then retransplant those. I think this is actually quite an interesting recent development in that field. And yeah, so that's in that sense, this was according to expectations. There's interesting to see also that you have the M1 and M2 polarizations in the macrophages that apparently attract equal amount of interest. But although I think for many people it will be hard to determine what polarization exactly is they want to target. This is also one of the questions in the podcast, of course. How to deal with those macrophages, and that becomes very clear from this poll that this is something that people are very eager to go into.
For me, it was also interesting to see which tumor indications people are working with or they're interested working with, of course, because that's more relevant for the native TME samples that we do receive in our team. And the usual suspects that I would say, the non-small cell lung cancer and breast cancer, melanoma, were the ones that scored more. But on the other hand, gastric cancers and CRC were also quite well demanded, let's say, in the assays, and other more rare types of cancers as well. And I think that's important to know. And unfortunately, we couldn't include even more options for this poll. There were only 9 possibilities, but there are many more that we can do in this type of assays.
Fantastic. Yeah, definitely. I mean, judging by our audience, we definitely do have a pretty diverse range of applications and fields that people are interested in. So out of those, for what types of therapies do you think these assays are most useful, do you think?
Well, I mean, there's really a broad range of applications that you can think of here. And it's, I think, nice also to step back a little bit and, at least comment on the 2D cell cultures and the organoid monocultures, because, of course, these are optimal models to sort of study biochemistry. If a protein binds to another protein, it doesn't really matter in what kind of 3D dimensions you will grow your cell. If those proteins are there, they will bind. So I think this is also the reason why many people still enjoy working with the 2D cell cultures. Super practical, very good for discovery, screening, cost efficient. These kind of development steps are really, something that fit very well within the 2D cell line space. But also, I think more and more evidence is there that we can, combine discovery and mode of action in the same screens using a bit more complicated models like the organoid models. And then, especially for the co-cultures that we have been presenting in our seminars, the co-cultures between myeloid cells, T cells, fibroblasts, these, I would say, are most suitable for, the mode of action studies. So not necessarily in the drug discovery steps of a drug development pipeline, but really, if you already have some promising leads based on screening in basic models, then these models become more relevant to study, because then the localization of the target that you want to engage with is heavily dependent on the 3D space and the culture conditions. Also, especially in upcoming fields like the antibody drug conjugate fields or other antibody-based approaches, they really require you to be able to see where is your antibody going. Is it binding to the right marker of interest? Are they delivering into the tissues? And how deep do these drugs penetrate into tissue? So this is really where the power of the 3D cultures and the co-cultures comes in. Speaking from my own expertise with the fibroblasts, they form physical barriers that you need to be able to bridge with your drug. They can metabolize drugs. They can, affect cells in their survival responses. So, in these cases, this Lego-like approach, like how I presented it, is very useful, because you can get your cancer of interest, you mix them with the fibroblasts, most of the properties are there that you want to study, and then you can really go into the biology a bit more. When we increase that complexity towards the immune system, this becomes a little bit more tricky. In seminar 2, Marit already very nicely demonstrated that you can make matched organoid immune cell pairs, where you go for autologous systems. Of course, the systems that I presented about in my talk, they are all allogenic. So, you will run into limitations that the T cell engagement with the cells. We can do very basic killing assays, infiltration assays. These kind of assays, they are still very suitable. But if you really want to go into this immune checkpoint inhibitor field, you need everything to actually be able to study this. And this is actually really, you know, where Natalia's platform with the ex vivo patient tissue comes in. So I think, you know, I would be out of my league to start mansplaining this. So go ahead.
Thanks, Martin. Yes, indeed. So if we are really speaking about recapitulating cancer immunity cycle in a dish, you know, in vitro, then in this case, the ex vivo assays would be our best bet, I would say, because this gives really a footprint of what is in there in this patient tumor, with all cell types that are represented there, including T cells and myeloid cells and fibroblasts and of course tumor cells. And it's all complex interplay that is necessary there to observe drug effects from some very complex drugs like immune checkpoint inhibitors. It is extremely difficult to see the effects in allogenic setups from these molecules, what Martin already mentioned. But in our system, in the ex vivo system, we do see effects from these drugs. And not only we can measure it by some cytokine increase, so indirect effect, but we also see direct killing effect of the tumor. And that's what makes this setup very valuable in the drug development process, because when you do have already a drug candidate and you need to test it in, let's say, real field-like conditions on a patient tissue, then this assay comes into play, and we can test on different indications and see if we see the response to this type of molecules like immune checkpoint inhibitors. And it will depend, of course, a bit on the patient and on the indication. But what we do see is that about 20% of the tissues that we receive respond to at least the known immune checkpoint inhibitors. And that also corresponds to what is seen in the clinic. So that's showing the translatability of this assay. But one of the downsides here is that we have to test enough patient tissues to see those 20% responders. So it's not like we can take one patient sample and hope that the drug will work on it. It means that we need to test 5, maybe sometimes 10 different patient samples to be able to see the drug effects of this kind of molecules.
Fantastic. Yeah, some really, really great points there. So thanks for that. So shifting gears a bit, so out of the different assays that was presented here, what stages of drug development do you think are most relevant for the assays?
I think what Martin already mentioned, that more simplistic assays, that do not involve a lot of cellular players, can be used at the earlier stages. When not for screening, but when the mechanism of action is needed to be known or traced a bit better in vitro. And regarding the ex vivo assays, it's really almost at the door, let's say, of clinical trials, where we can test 1 or 2 best candidates compounds in a patient material. So think about it a bit like in vitro clinical trial setup.
Yeah, that's absolutely. I can really adhere to that. It's for especially, I think the reconstituted TME approaches are really on the crossroads of academic discovery and the actual mode of action studies, especially in the high content imaging platforms. We can really study in a lot of detail how your drug is affecting a tumor. But indeed, this is really also dependent on the drug types, on the type of screen you want to perform. Because I think both the ex vivo platform and the reconstituted TME, they are quite time consuming and labor intense procedures, and so this makes these procedures also a little bit more expensive to perform. So it's always better to already know a little bit what you want to look for, because unbiased screening in these kind of setups is really not very feasible or cost effective, I would say. So it's all, I would say if you can put it somewhere in the drug development pipeline in the year 2 or 3 after you have your lead compounds, and then are really eager to move on.
Perfect. Yeah, great points all around. So yeah, again, Martin and Natalia, thanks so much for joining us today. It was really nice speaking with you. Thanks everyone for tuning in. If you enjoyed this episode, be sure to check out our recent webinar together, and you can also catch the next webinar in our Cancer Immunity Cycle series starting on March the 2nd, which will be called What's Next in Preclinical Cancer Immunotherapy Research. That one will be a panel discussion covering new and emerging methods and future directions for immuno-oncology. We hope to see you there, and have a great day, everyone.